Hello and welcome to BioRules video lessons. Today's class is going to be short and sweet, but stupidly important. So, pay attention. Condensation. Condensation is when we put together molecules, when we make two molecules react together and make one. So here I'm going to be representing one molecule. Take your time, tell me which molecule is this. Okay, if you guessed the glucose, well done. This is a very good guess. It could be a galactose, but don't care. For us here, this, when you see something like this, it's a glucose. 6 carbons, making an hexagon, one side branch, that's it. And which one is this? Take your time. Yes, if you said the glucose again, yes, we have two glucose over there. And what you're going to be doing is the condensation of these two molecules. So we are going to be putting these two biomolecules together. We're going to be making them to react and becoming one bigger molecule. But for that, we are going to be moving for the next slide, where I'm going to be showing you the condensation again of these molecules. But I have removed some of the hydrogens for us to be able to visualize things in an easy way. So what's going to be happening here is this. First thing, to do the condensation, I am going to be separating this here. See, I separated one of the hydroxyls, OH, I put there on the corner. Hey, there it is. And I'm going to be separating from one of them here. I'm going to separate the hydrogen. There it is, I separated. And I put it together with the hydroxyl and I made water. What we have there in the corner is H2O, we made water. And what I have here now, there's a carbon there that needs to bond to someone. That carbon is going to be bonding there. So what we have here is a monosaccharide, another monosaccharide, and they are bonded together here with an oxygen in the middle of them. We have now a disaccharide. This molecule is called maltose, two glucoses bonded together by condensation. Okay, so this is how it works. Another example for you. So first thing, which molecule is this? Take your time. Yes, if you said glucose, you got it right. And which molecule is this? If you said ribose, you got it wrong. Actually, this is a fructose, one carbon over there, carbon, 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 and another carbon over there. So this has 6 carbons making a pentagon. This is a fructose. And we reacted it together, so OH with OH over here. This makes water. So over here, water will be made. And we have here now a glucose connected to a fructose with an oxygen in the middle. So we have here now an oxygen that's connecting both of them. It's like now the glucose and the fructose are holding hands. So there it is. And here we have another view, glucose and fructose making sucrose, same thing, having a sucrose, glucose and fructose holding hand here in the middle. This is condensation. Okay, a few more examples. On the top we have glucose with glucose holding hand, making water, and we made maltose. And in the bottom we have a glucose and a fructose reacting condensation. They are releasing water, now making a disaccharide called sucrose, and they are holding hands. So this was condensation. For you to remember what condensation means, there are two things that happens in condensation. You are putting two compounds together, so you are making them to get close, like if they are more dense, more condensate. So this is one way for you to remember what condensation is. And another thing is that when you bond them, you make water. Water is produced. And similarly, when you take a water bottle from the refrigerator, water shows up on the surface because of condensation. So no matter what you're going to be using to try to remember what condensation means, condensation is this, putting two molecules together and making water. Okay, now we'll talk about hydrolysis. Hydrolysis is the opposite of condensation. Simple as that. If you think about condensation as putting two biomolecules together making water, hydrolysis will be using water to break two to break up biomolecules into biomolecules separated. Okay, so this will be hydrolysis. So I have here maltose that was made by condensation a few moments ago, and what I'm going to be doing now is to be using this water over here to be able to hydrolyze, to break down this maltose into glucose. So the first thing I'm going to be doing is to separate it. So here we have go for one side and separate here, go for the other side. There they are. They have been broken. So now what do? I get one hydrogen to put with that oxygen to make a hydroxyl on the left, and the other hydroxyl from the broken water comes here and is put over there. I just made hydrolysis. I separated and I reverted that disaccharide to the two monosaccharides that make it. Simple as that. An example here for you: sucrose, glucose on the left, fructose on the right, holding hands. And if I put water and I put an enzyme called sucrase, for you to make these reactions we need enzymes. We put an enzyme here called sucrase and I separate it into glucose on the left and fructose on the right. Simple as that. This is the hydrolysis process. Okay? I hope you have enjoyed. Short and sweet, as I told you, literally sweet, because we're talking about condensation of sugars, of carbohydrates. And now I have a quiz here to make sure that you understood everything. Have fun. Bye-bye.